My name's Sam Smith. I'm in the research management team. Here today, joined by Rachel Lennon, Dr Tzehow, David Sherwood, and Eamon Sheridan, who will hopefully be chairing in a moment. Eamon is the chair of our GSIP board, and I will give him hopefully a chance to introduce himself later. Our speakers today, Dr Tzehow, a consultant neurologist and senior clinical fellow at NHS National Prion Unit and MRC Prion Unit at UCL. His presentation will be evaluating the penetrance of PRNP gene variants, by example. And then straight after that, we'll have a presentation by Rachel Lennon and David Sherwood on defining basement membrane. I will introduce, and hopefully Eamon can introduce them later. If anyone has any questions throughout either of the presentations, please use the Q&A function in Zoom, and we will answer the questions after each talk. There'll be a few minutes to answer each question, and we can type any answers that we can't cover that we don't have time to speak about. We also have our next research seminar is in November. We'll be joined by Liz Ormondroyd, talking about additional findings on the 100,000 Genes project, and Eleanor Scrooby, who will be talking about gene-to-patient approach for gene discovery. And that will be shared by Julian Hazensford, chair of our participant panel. I'm going to pass over now to David. I think is going to share his screen.
I thought we're not going first. We're going.
Oh, sorry, so Mok. Sorry.
Yeah.
I'll go first.
Okay, let's try. Oh, no, it's maybe this one. Is that sharing properly?
Yes.
Great. So, well, thank you very much for inviting me to the seminar and to speak about how we evaluate the penetrance of prion protein gene variants in the National Prion Clinic and the MRC Prion Unit at UCL. I think the initial invitation was to talk about our recent paper on characterization of the 2 octopeptide repeat insertion, but we can't really talk about that without talking about another mutation on which we've sort of first did the work on, evaluating how we sort of evaluate whether a new or a variant is penetrant or pathogenic. And even before that, I think it would be useful to just go through a few key aspects of prion biology, because it will inform us. It will inform the techniques that we use that I'm going to show to determine these 2 aspects. So, how do I? Oh, there we are. Right. So. The prion diseases are also known as transmissible spongiform encephalopathies. It doesn't only affect humans, and you can find it across several species. The oldest described prion disease is actually scrapie in sheep and goats, going back a few centuries. Now, in humans, the commonest form of prion disease is sporadic creutzfeldt-Jakob disease, which is sporadic CJD, and that accounts for about 85% of cases. Inherited prion disease, which we're going to focus on, usually accounts for about 10 to 15%, and we have the very rare acquired prion diseases such as Kuru, variant CJD, and iatrogenic CJD. So this is a picture of what our normal prion protein is supposed to look like. You see the helices, and you see a bit of the protein that anchors it to the cell membrane. But importantly, you see these 3 sort of grape-like structures, which are sugars at positions 181 and 197 in the human prion protein, and they do play a role in a molecular strain typing, which we'll discuss later on. And the way prion prions sort of cause disease is fairly unique. It's due to a misfolded disease-associated conformer of the prion protein, which has the ability to recruit the host-encoded normal prion protein into amyloid, and it elongates, and then it fragments or breaks down, and it basically initiates this chain reaction. And it is completely only dependent on the protein. It does not require any DNA at all. So so I thought perhaps we should look at the prion protein gene first. And this is just a schematic representation of all the gene variants that we know of. Now, this is not up to date, and there are a few novel mutations we've recently discovered that we're preparing manuscripts on, and will hopefully be out within the next year. And you can see already that these various variants within the prion protein gene can either be, highly penetrant disease-causing mutations that run in families, and some are, a bit uncertain. And the two that I've circled out here in the red squares are the ones that we're going to discuss. And it was at the time when this schema was, this diagram was conceived, they were thought to be possible causes of familial CJD, but hopefully I'll be able to convince you that later on that they're perhaps merely a low risk factor, or possibly even a benign variant after all the work we've done on it. And I think the other bit that really interests me is that there are some variants that can confer resistance to prion disease. For example, the G217V was discovered in the Kuru affected population, which gives you complete protection against that condition. And also heterozygosity at codon 129 confers a bit of resistance to various forms of prion disease as well. So the canonical clinical phenotypes of inherited prion disease, the well-known ones are GSS, which usually associated with the P102L mutation, and this causes a chronic ataxic syndrome with pyramidal features for several years before onset of dementia in the later years, and patients usually live for about 4 to 5 years before succumbing to the disease. And of course, there's this familial fatal insomnia, which is, I've seen a couple of patients with these, and it's a really awful, awful disease. If you're one of the old school physicians like me who have gone through 48 or 52-hour shifts with no sleep, you can just imagine what it feels like to have FFI. And then finally, we've got this familial CJD, which is clinically, radiologically, and even sometimes neuropathologically indistinguishable from sporadic CJD. And so they present with dementia, myoclonus, ataxia, and are usually dead within 6 months to a year. But more recently, we've sort of characterized a different type of inherited prion disease that presents with peripheral amyloidosis. So these patients, this is a truncation mutation that causes an anchorless prion protein, which has the propensity to form amyloid, and it can be deposited in multiple organs. And these patients can have chronic diarrhea for decades, followed by dysautonomia and peripheral neuropathy before developing dementia. So some of them could be lingering in sort of GI clinics somewhere in the country. And of course, there are the others, such as the octopeptide repeat insertions. And the interesting thing about OPRIs, or the larger OPRIs, is that they can either present like familial CJD, again, indistinguishable from sporadic CJD, or some of them present with these long duration frontotemporal apraxic syndromes that can last 2 decades or more. And, most of the time in the highly penetrant mutations, you've got a fairly characteristic clinical phenotype, you've got a family history with an autosomal pattern of inheritance, and you've got, pretty characteristic features on neuropathology, and it's, a closed book. These are highly penetrant mutations. But then occasionally we find, gene variants, usually unexpectedly, that don't either have no family history or no distinct neuropathological features. And the question then arises whether, are these pathogenic mutations? And if they're pathogenic, what is their penetrance? And it is in these situations that we try to harness multiple lines of evidence, including molecular strain typing, looking at large-scale genomic data databases for the variant allele frequency, and also considering the prion protein structural perturbations to give us some idea as to whether these are penetrant or not, and if so, how penetrant. Now, I've listed in silico predictions and with a strike through because these are fairly useless in prion diseases, and I'll show you why later on. So, and in neuropathology, when we look at a brain of a patient with prion disease, we see spongiosis, which are holes in the brain, which you can see up here. And there's also reactive astrocytosis, neuronal loss, and if you stain them with anti-PRP antibodies, you might see plaques, or this is what we call sort of synaptic pattern, and occasionally they deposit as amyloid in this picture. And when you see PRP antibody staining like this, these are called multicentric plaques. You find these are quite characteristic for inherited prion disease of a certain type. The other characteristic pattern that we see, which I have not shown here, is called cluster plaques, which are pattognomonic of variant CJD. So neuropathology gives us an idea of what type of prion disease this is. And in inherited prion disease, particularly in the mutations in the N-terminal end, we see these threads here that you can see pointed up by the red arrows in the subcortical white matter that look like filaments. They are present in point mutations towards the carboxy terminal, but they are less frequently seen. And in particular, if you have in octopeptide repeat insertion, and this is a picture of the cerebellum, you see this striped pattern of PRP antibody staining that resembles either, depending on what type of animal you like, a tigroid stripe, or for me, it just looks like a brindle greyhound. And the next thing we want that gives us a lot of information about prion disease is molecular strain typing, and we employ two techniques which are interrelated. One is Western blotting and the glycoform ratio. Basically, when you have frozen brain tissue, usually frontal cortex, from a patient with prion disease, you homogenize it, and you can partially digest that with proteinase K, and it generates usually three distinct fragments with different motility on the gel, and the motility is dependent on whether it has 2 sugars, 1 sugar, or no sugar. So diglycosylated, being heavier, moving slower, is this. Monoglycosylated, second band, a bit lighter, moves a bit faster. Unglycosylated, no sugars, moves very quickly. And there are two different classifications that we use. In the UK, we use the London classification at MRC Prion Unit, and and in CJD there are 3 types, and type 4 is variant CJD, and you can see the different intensities of the glycosylated or unglycosylated bands, and they have patterns that are specific to the disease. Worldwide, there is a Parker classification in which the samples are processed slightly differently, but it gives you also 3 bands. And if And what we can also do is once we've blotted the brain, we can analyze the predominance of the glycosylated fragments, and they are different for sporadic CJD versus PRNP point mutations. You can see that in the PRNP point mutations, classically P102 or D178N and E200K, the diglycosylated fragment predominates, while in sporadic CJD, it's the monoglycosylated fragment. And on the far right of the screen, we see the glycoform ratios of the octopeptide repeat insertions. And the thing about this is that it is more or less indistinguishable from sporadic CJD. So you can't really use glycoform ratios to distinguish between sporadic and OPRI, but you can use it to distinguish between sporadic CJD and point mutations. So the story now begins with actually T201S. And this is a patient, and it started with a patient that I encountered in the southeast of England, and she basically presented like sporadic CJD, really rapid disease, dead within 8 weeks, with an MRI scan that looks like sporadic CJD. And you can see here that in the diffusion-weighted images, you can see high signal in the caudate head and along the cortical ribbon, with corresponding low signal on the ADC map and also in the cortical ribbon. And this is what we see in sporadic CJD or familial CJD. So once it's one of the easiest neurodegenerative diseases to diagnose, because once you see this, the diagnosis is more or less, CJD. Now, and so, told the family, this is sporadic CJD, there's no family history, but we will take some blood to sequence the prion protein gene just to be sure that this is not familial CJD. And we discovered this T201S mutation. So we're like, oh, never heard of this before. And it was not, what was the first thing we did? Well, we Googled it, and we found a little abstract that was submitted to a stroke conference, which is a Danish case from 2009, and the history and the clinical features are exactly, very similar. It's CJD, but you found a T201S mutation. The thing about this is that they actually did an autopsy here, and we had brain tissue to analyze. So we contacted our Danish colleagues, and they very kindly sent us some brains, and we stained them here. And so you can see on the top panels, it shows spongiosis. There's little holes, it looks like a pink sponge. And when you stain it with ICSM35, which is a PRP antibody, you see this sort of a diffuse synaptic labeling. So again, indistinguishable from sporadic CJD. So then what did we do? We thought we'd look at the protein structure. So the only difference between threonine and serine is a methyl group, and this is in the solvent exposed part of the prion protein, and not in the protease resistant core. So we thought perhaps this might not perturb the structure too much and destabilize it. But then another way of looking at whether a variant is likely to be penetrant or pathogenic is to look at the in silico predictions. So we put it through 3 well-known tools, and it came out that polyphen-2 thought it was possibly damaging, CIF thought it was deleterious, and the CAT score was 26, which ranks it within the top 1% of pathogenic variants. However, when we put the other known benign and highly penetrant prion protein gene variants through these tools, the outcome was mixed. So overall, we felt that in silico predictions are not particularly useful for calling pathogenicity in PRNP variants. So what did we do? We had brain. The brain looks like sporadic CJD. So we blotted it and we analyzed the glycoform ratios. So this figure on the right, the left hand corner here shows the glycoform ratio of T201S, which corresponds to sporadic CJD. And it's different from what we see in point mutations such as E200K. So the ground is now shifting. It's starting to look like T201S might be either bystander, a neutral variant, or it's very low risk. And the CJD in these ladies' condition may be just sporadic. So what else can we use to support our formulation? At around the same time, Eric Minikel and his group published a worldwide collaboration on quantifying a prion protein gene variant penetrance. And within this, you could calculate the penetrance by using the lifetime risk of CJD, which is 1 in 5,000, multiplied by the variant allele frequency associated with disease divided by the occurrence of these mutant or variant alleles in the general population. So we interrogated the Broad Institute's GNOMAD database, and we found one other allele. So by applying this formula, we had a central estimate of 0.46%, but because this is very close to 0, and to make it meaningful, we assume that this is a binomial distribution and use the Wilson score interval to calculate the lower and upper bounds of the 95% interval so that it doesn't cross 0, which makes it meaningless. So the worst case scenario from our calculations is that the upper bound of the 95% confidence interval is 9.5%. And this lands it squarely there with a central estimate of less than 1%. And of note, these highly penetrant mutations, E200K, P102L, and D178N are not found in this in the GNOMAD, which is consistent with what we would expect. So overall, using these lines of evidence, we conclude that this is most likely a low risk or benign variant. And as such, we felt that it is below the threshold to justify predictive testing in at-risk individuals. And it so occurs that the lady whom I saw had a daughter who was really insistent on having predictive testing, and we were able to use this the evidence that we've garnered to reassure her. We also referred her to the local clinical geneticist who independently came to the same conclusion as we did. So it turns out that all these sort of avenues of evidence, information are useful clinically. So now, coming to 2-OPRI, which is a more recent paper published in Viruses last year. Now, this was led by our US colleagues because they had the most number of cases. And it rose because, again, I saw a patient who had sporadic CJD. And surprisingly, when we sequenced this prion protein gene, we found a 2-octopeptide repeat insertion. And he did not have an autopsy. But then I remembered that in the previous paper published by Hill et al. In 2006, from which we used the glycoform ratio, there was a case of 2-OPRI included. And this case dated, so we did a bit of digging, and this case dated from 1985. And this man presented with clinical picture similar to sporadic CJD. He had a slow EEG and had a 2-OPRI insertion in the prion protein gene. An autopsy was done, but we were unable to stain the fixed paraffin blocks, probably because it was too ancient. However, importantly, there was some frozen tissue archived, and we were able to do further from that. So at So just to give you a quick background about octopeptide repeat insertions, the octopeptide repeat insertion region in the prion protein gene is between codon 51 and 91, and it's composed. It starts off with a nonapeptide, which is 9 peptides long, and then followed by 4 octopeptides. So anything more than that is an extra opry. So when we when we say someone has 4 opry, for example, we really mean that they have 1 nonapeptide and 8 octopeptides with 4 extra. And when we say someone has 2 octopeptide repeat insertion in the prion protein gene, it's a total of 1 nonapeptide, 4 normal octopeptides plus 2 additional ones. And we actually know a great deal of octopeptide repeat insertions in prion protein gene between 4 and 6 opry, because they're very well characterized. And in fact, it was the 6 opry family is one of the first familial neurodegenerative diseases characterized in the UK back in the early 90s. Now, what about 2 opry? We don't really know much about 2 opry because there's limited information from the literature. There are definitely 2 cases associated with sporadic CJD phenotype, and there was one found in a patient with a long duration dementing illness who was not autopsied. So we don't know whether that patient actually had prion disease. And in one family, there are multiple carriers who are asymptomatic. So at the prion, I think it was 2018, we bumped into Brian Appleby from the US, and he had 5 cases. So he then led this intercontinental collaboration from which we garnered a total of 8 cases worldwide, and from which 5 US cases had autopsies available, and the one that we had in the UK, although it was too ancient for immunostaining, we did have frozen tissue, and the female to male ratio is 1:1. The age range is squarely in the sporadic CJD peak range, and the disease duration between 2 and 21 months is consistent with sporadic CJD. And the codon 1 to 9 that we have are a mixture of MMs and then Vs. And interestingly, from this group of 8 individuals, there are 4 blood relatives also with long duration dementia, but from whom the prion protein gene is not sequenced. Now, we'll come back to this because I'm slightly uneasy about this because we just don't know what they have. They could have, we'll come to that later. So first thing we do.
Mark,
yes.
we're at 2:30 already.
Oh gosh, sorry.
You may need to kind of speed along.
Right. So neuropathology looks like CJD. Nothing that looks like familial inherited prion disease. The Western blots look like CJD. Okay, and then as we know, the glycoform ratio is not particularly useful, so this is not discriminative. But then we had the large-scale genomic databases that we can interrogate, and one of it is the 100,000 K Genomes Project. So overall, the central estimates are about 0.2%, and the upper bound of 95% interval is less than 2%. So our conclusion is that this is possibly at most a low risk factor, which increases your lifetime risk if you have this insertion from 1 in 5,000 to 1 in 500. So the caveats are inevitably the numbers are small. 2, we lack the autopsy data for the carriers or at-risk individuals with the long duration dementia, so we don't know what they have. So if they turn out to have prion-related neuropathology, this changes the picture completely. And thirdly, the databases that we interrogated are genomic databases and not normal population databases. And especially with the Genomat, they come from, some of them come from neurological illnesses, so they're not completely non-neurological. And of course, in some of the genomic databases, the probe length is 150 base pairs, but in 2 opries, your base pair number is 179. So there's a possibility that we're underestimating the allele frequency in these databases. And for very rare variant alleles, such as T201S, we might be underestimating the penetrance. or the upper bound of the 95% interval. Because if you expand the denominator by 10, 10 times, you might actually raise the upper bound of the 95% confidence interval to about 90%, which makes it a bit meaningless, really. And the last thing is that outside of a characteristic family history and pathology, there's no single line of evidence that dominates. And this is just our current practice at the National Prion Clinic and the MRC Prion Unit. And we're still learning and evolving, so things may change. And finally, what is the future? The future is this. We're now able to visualize at near atomic level what the prion protein, what prions look like. So this could completely change the way that we look at these things in the future. So sorry about running over time. Thank you very much.
Thanks, Mark. We haven't unfortunately got time for any questions, but we'll zoom straight on to Rachel and David. So if you guys want to fire away, that'd be brilliant. And I'm sorry that your time is probably a little shorter than you were hoping, but I'm sure they'll be able to manage. David, thank you very much.
No problem. We're just sharing the screen. So thank you very much for the invitation to present this collaborative project on basement membranes. These are supramolecular structures, and we hope to convince you that they have vast unexplored human disease associations. So I'm Rachel Lennon. I'm a paediatric nephrologist and director of the Wellcome Centre for Cell Matrix Research in Manchester. And I'm presenting in duet with David Sherwood, who is not a gastroenterologist. He's assistant chair of biology at Duke University, so he's joining us from North Carolina. So basement membranes are in all of our tissues. They surround muscle, they underlie epithelia. Dave, we just need this. Oh, there we go. And they are sheets of extracellular matrix protein. So you can see in the scanning electron microscopy image here, the sheet-like structure of the basement membrane with the epithelia separated from the connective tissue underneath. Basement membranes are a defining feature of all multicellular animals, and they have increasing associations with human disease. So in my lab, we focus on basement membranes in the kidney. And so what you can see in the movie on the left, this is 3D electron microscopy images going through a glomerular capillary, which is where blood filtration happens. And the dark structure, the ribbon-like structure, is the basement membrane, and the yellow arrows are highlighting defects in this basement membrane. These defects allow cells to cross from the blood out into the urine. You can just see this red blood cell crossing across this defective basement membrane. And over time, these basement membranes in the kidney filters, they thicken, split, and lose function. And this happens in a number of genetic kidney diseases. And one of the most common that we see is Alport syndrome, which is caused by variants in the type 4 collagen genes, COL4A3, 4, and 5. And this leads to abnormal basement membranes, and as well as causing a fairly major kidney phenotype, there's sensory neural hearing loss and ocular problems. So how do we study basement membranes? Well, we can use a range of experimental systems. We can use cells in culture. All adherent cells in culture will secrete and assemble basement membrane proteins. So we can learn a certain amount from cells in a dish. We can look at more complex structures, such as organoids, and use stem cells to differentiate into kidney organoids. And these structures pattern beautifully into the filter and tubular structures that we see in the kidney. But this doesn't tell us about physiology. For that, we need animal models. And the zebrafish is a really good model to look at kidney physiology. The embryos have a single midline kidney filter. And this allows us to study physiological function of basement membranes in the kidney. And we can go to the mouse if we want to model human disease. But all of these systems have their limitations. And for this project, we wanted to really embrace basement membranes in a broader sense. And so we teamed up with Dave's lab. So I'm going to hand over to Dave to introduce his system.
Thanks, Rachel. Yeah, in our lab, we use C. elegans to examine basement membranes. C. elegans are an incredibly powerful model. They're optically clear, so we can visualize all the tissues in vivo for the lifetime of the animal. It has powerful genetics and RNAi, so we can knock down genes conditionally at any time during the animal's life cycle. And also it's very amenable to transgenics, and we've used CRISPR-Cas9 to knock in neon green and other fluorophores to over 60 basement membrane proteins, so we can actually look at the living matrix in an animal. And with this toolkit in this system, we study a lot of different aspects of basement membrane biology. Just a couple of examples. We look at how basement membranes emerge in development. The first component that emerges and enwraps tissues is laminin. It's associated with the cell surface. And you can see that in the center panel here, emerging and enwrapping in all tissues during embryogenesis. We can also examine how cells invade through basement membranes as a model for metastasis. We have the only model in the world where we can actually watch a cell in a living animal breach a basement membrane to understand how cancer cells hijack this program. And C. elegans also age very rapidly. They only live for about 2 weeks, and their basement membranes age just like ours. This is a knock-in of type 4 collagen, a core component of basement membranes. And just like our tissues, type 4 collagen builds up dramatically on tissues over time, compromising tissue function. So we can examine how this occurs. So a number of years ago, a C. elegans biologist and a pediatric nephrologist found themselves sitting next to each other, at a Gordon Research Conference, and wondering why all schematics of basement membranes just showed a few molecules. Here we have the laminin that I just talked about that associates on the surface of cells with integrins and dystroglycan to help them polymerize. On top of that is another network of type 4 collagen. This network is cross-linked together with covalent bonds and bestows basement membranes of tensile strength. And then there's inevitably a few other components like perlecan and nitogen shown that cross-bridge these two networks. And yet Rachel and I started talking, and there were hints in the literature, and particularly her proteomics, that suggested basement membranes could be vastly more complex and have upwards of 100 different components. And the problem is that basement membranes are insoluble. Many of the components are insoluble to biochemistry. They're difficult to purify. You can't just do a microarray and ask, well, what's in a basement membrane, because it's outside of the cell. And what really needs to be done is the painstaking approach of looking at molecule by molecule, every single component, and does it actually localize to a basement membrane in vivo. And that's the genesis of this paper here, this collaborative project, where basically we wanted to find what is the parts list for a basement membrane so that we can really understand their complexity and function. And we were aided in this journey with two remarkable postdocs, Michael and Ranjay, and we assembled an initial pipeline of the most ambitious number of basement membrane components, 103 from gene ontology. And then we used a number of other strategies to query and add potentially other components to that list, things like a basement membrane signature in C. elegans, protein domains that are enriched in basement membrane proteins, localization studies, and biochemical interactions. And this led to an expanded list of over 260 basement membrane zone genes. And then we painstakingly went through the literature and also genome edited and tagged many of these components in C. elegans to ask, do they localize to the basement membrane? And we came up with a confirmed list of 188 and an additional 34 components that biochemically strongly interacted with other basement membrane proteins, or their enzyme activity acted on basement membrane components. And this led to an astounding number, 160 basement membrane matrix proteins and 62 transmembrane proteins. So this is the most, I teach cell biology, this is the most complex supramolecular structure that I know of. And this suggested that this basement membrane may have many additional functions. And we were very interested now with this parts list to understand more about basement membranes. And so one of the things we did is we examined all of these proteins and we looked at the different domains that are present in them. And then we created an interaction network. And what you can see in this interaction network is a lot of proteins grouped together, like the Atom TS proteases, the matrix metalloproteinases, the integrin receptors. These are proteins that share similar domains and that thus cluster together. But notably, we found some proteins that seem to bridge multiple different islands. One is papillin, which we'll come to at the end, a very poorly studied but highly conserved protein. Another one is HSPG2. This is perlecan. So these proteins share many domains with different groups of proteins, suggesting that they could have important regulatory functions. So we wanted to test this, so we looked more closely at perlecan. And the problem with perlecan is knockouts in mice are lethal and knockouts in C. elegans are lethal. So we took advantage of conditional RNAi, and what we noted in C. elegans is that this perlecan localized to the gonadal basement membrane. This is the basement membrane around the gonad with the germline and oocytes, but only after the gonad had formed. And so we used conditional RNAi to begin knocking down perlecan in the early larval stages when it's growing, and this had no effect on the basement membrane. However, when this gonad fully formed, and we're just looking at one of the gonad arms here, what you see compared to wild type, and here we're looking at that early basement membrane component laminin, is that this is a scrunched and starry looking gonad, and the laminin is bunched up in fibers. And this is also true for collagen, another core component, as well as nitrogen, where it looks like in the absence of perlecan that this basement membrane is not organized correctly, falling apart, and the tissue misshapen. So this really shows that we can begin to identify key regulators of basement membranes with this network analysis. Another thing that we did is we took advantage of the rapid ability to knock down genes in C. elegans, and we did a screen knocking down every single basement membrane component. And just highlighting one of our findings, what we found is that signaling pathways whose ligands localize to the basement membrane, TGF-beta and SLIT, both of whose ligands localize to the basement membrane, when you knock out these pathways, there's feedback such that TGF-beta signaling, for example, not only does the ligand localize to the basement membrane, but the signaling itself affects the composition of the basement membrane. And this is conserved when we knock this out of zebrafish. What we also found by pharmacologically knocking out the TGF-beta 1 receptor, here's the trunk region, a cross-section through a zebrafish, normal levels of collagen. There's a doubling in the levels of collagen. So this was really fascinating in that signaling pathways might be able to tune and regulate basement membrane composition. Well, we next wanted to explore, now with this expanded list, the disease association, and I'm going to hand it back over to Rachel.
Yeah, sure. So we first asked how many of these genes have associated human phenotypes. And so what Anna used was the OMIM and human phenotype ontology databases, and found that 112 had associations already. And as you can see on the right part of this first diagram, 91 of those had definitive evidence of causing disease in the Genomics England panel app. So the phenotypes, in terms of the range of phenotypes affected, then this spanned 24 top-level HPO terms. And what this does is just highlight the relevance of basement membranes across a broad spectrum of human pathologies. But the next thing that we wanted to do was look for new disease genes or disease modifying genes. And for that, we used data from the 100,000 Genomes Project and teamed up with Jamie Ellingford, who screened through over 63,000 whole genome sequences and identified over 5,000 unique and rare predicted loss of function variants. So we needed, in amongst all of that, we needed to focus somewhere, and we focused in on 40 genes where both alleles were affected. So either homozygous or compound heterozygous variants. And then with further filtering, we got down to 28 genes with high penetrance. And in amongst that group, there were those with green level or definitive evidence existing for disease causation. And many of those we were familiar with, the collagens and laminins. But a number of these genes had moderate or no evidence of prior association with human phenotypes. So we selected 3 of those for follow-up functional studies. And in the next three slides, I'll just run through the studies that we did for each of these genes. We started with matrillin. So the first gene encodes matrillin-2, and we found 3 individuals with predicted loss of function and overlapping phenotypes. So matrillin-2 is a relatively recent gene, and there are no orthologs in C. elegans or zebrafish that we could use. But it is expressed in human kidney cells. And so what we were able to do in this study was use CRISPR to knock out matrillin-2 in the kidney cells, and then with lentiviral transduction, we were able to rescue with the wild type or introduce the variant coding sequence. And what we found was that the variants resulted in abnormal matrillin-2 secretion, but furthermore, with proteomics, we could see that in the absence of matrillin-2, the composition of the basement membrane, or the matrix that these cells assemble in vitro, was abnormal. So that's giving evidence of a functional effect of those variants in matrillin-2. For the next gene, MPZL2, this was already associated with a sensory neural hearing loss phenotype, so high evidence, definitive evidence of that association. But we had observed in the individual we were following in the 100,000 Genomes Project a kidney phenotype, and I should say that sensory neural hearing loss is associated with abnormal basement membranes. So we wanted to pursue some of the functional studies, and for this we were able to use zebrafish because there's an ortholog for this gene in zebrafish. And what you can see in the bottom left is abnormal development of the tail. So this is reduced fin fold extension in the CRISPR knockdown. And then when we looked in the at basement membranes in different organs, we could see actually that was fairly intact in the gut, so wasn't affected by MPZL2 knockdown in the gut, but the kidney basement membranes were abnormal. So the red arrow here at the bottom right, just highlighting an abnormal region of basement membrane. So evidence there that MPZL2 loss disrupts certain, not all, but certain basement membranes. In the last example, we focused on a laminin gene. So as you've heard already, laminin networks are crucial for basement membrane assembly and maintenance. Yet this gene, laminin alpha 5, does not have strong evidence of disease association. In this particular example, we were able to pursue more detailed studies as the family was local to Manchester, and so we were able to access patient samples. And what we observed in this particular family was an early onset multisystem disorder affecting, causing mid-gestation fetal loss. And the pathologies included absent or abnormal kidneys. So with a mini gene, one of the variants here was a splicing variant. So we used a mini gene assay to confirm that that was had a functional effect. And then we looked at tissue. So post-mortem tissue confirmed an abnormal cystic kidney. And with focusing in on the laminin signals from the basement membrane, we were able to see that this compared to kidney at an equivalent stage of development, so 21 weeks, where we see nice laminin distribution in basement membranes. This was very abnormal in the 19-week kidney that we examine. So evidence here with the laminin alpha 5 variant of abnormal basement membrane assembly. So overall, what we have established in this project is a discovery pipeline for basement membrane research. And we've curated this information into a web resource that's basement membrane base. And as you've heard snippets of the story today, but we've identified new regulators and human disease associations. And hopefully we've indicated to you the value of using complementary experimental systems from C. elegans through to human cells. So where does this go next? Well, I'll hand back to Dave to explain first.
Sure. Thanks, Rachel. So one of the things now with being able to identify key nodes, we can rapidly go through them and see what their functions are in basement membranes. And one of them that I hinted at earlier is papillin, which is highly conserved but incredibly poorly studied. When we knock it out of C. elegans, it results in early larval lethality and very misshapen organs and a threefold increase in type 4 collagen. And this is really fascinating and potentially important because collagen builds up during aging and in many different diseases like Alzheimer's and diabetes. And we're now also looking at what is the function of these signaling pathways that regulate basement membrane composition? Is this a way that during development, normal physiology, and disease, that the composition of basement membranes can be altered? Back to you, Rachel.
Yeah, so what we want to know is whether there are basement membrane biomarkers of tissue health that could help us determine whether basement membranes are intact in early or late stage injury. We also want to know how to fix the problem. So can we use our knowledge of basement membrane biology to design strategies to reprogram basement membranes? And that could be with gene targeted or protein-based interventions. So with that, I'll conclude and acknowledge the wonderful members of our lab who have contributed to this work, so in my lab and in Dave's lab, to our collaborators, including Genomics England, and to our funders. And I'll thank you for listening.
Thank you much. Thank you very much, Rachel and Dave. That was a really fascinating demonstration of the use of the interaction of genetics and model organisms, which kind of we all talk about, but you presented it so succinctly that was really nice. Thank you. There is a question from Shisheng Guo, who says, Shisheng, I don't know if you want to unmute. Can we unmute Shisheng so that the question can be asked? Let me just see.
Oh, I can see it.
Okay. Can you see it?
Can you see it? There are. We just published this paper this year, and there are already 3 citations that are using the basement membrane network as markers for cancer progression.
Yeah, so
absolutely.
Yeah, Shisheng's question was any pattern to the role of basement membranes on cancer progression. Says thanks in response to that. Thank you very much, Dave. That's tremendous. I don't know if anybody had any questions for Mok that they wanted to ask. We didn't get the chance to ask him. There's no further open questions at present. Well, thanks ever so much for that. That was really great. I apologize to absolutely everybody for having screwed the entire thing up.
We can hear you now. That's good.
Yeah,
we can't see you though.
I'll put the video on. I was most amused by Mok's comment about him being an old school clinician. And I was just thinking, hey, Mok, listen, mate. Compared with me, like you're not even an old kindergarten clinician. But anyhow, thank you ever so much. So I'd like to thank all of our speakers and the long-suffering staff at Genomes England for hosting this for me and setting me up. Thank you ever so much to everybody. It was a really terrific series of talks. Thank you ever so much. With that, just to remind everybody about the meeting in November, the last Tuesday, 29th, I think it is, hosted by Gillian Hastings. Really great to see everybody there. as Sam said at the beginning, I'm the new chair of the GSIP board. So unfortunately, you will be hearing more from me, hopefully more efficiently in future. Thanks ever so much, everybody now. Cheerio. Bye-bye.